Hello, I'd like to thank everyone for watching the rebroadcast of our webinar that we held on June 22nd. The title is Opportunities for Using Nutrition to Improve Poultry Production. We have Dr. Elizabeth Bobeck. She is an immunologist at Iowa State University. She has her PhD from University of Wisconsin-Madison, and she completed a postdoc in applied nutrition and then a second postdoc in immunology before she joined Iowa State as an assistant professor of animal science in January of 2016. So without further ado, Dr. Bobeck, please go ahead and present your webinar, and thank you for joining us.
Yeah, thank you guys for getting this rebroadcast together so we could let others access or re-access the program. So today what I'm going to be talking about are different opportunities for using nutrition to improve poultry production. And our focus will mainly be on laying hens, but of course some of this is also going to apply to broiler birds. So today what we'll be covering are different topics involving eggshell quality, omega-3 supplementation in laying hens, the value of distillers, grains in layer rations, and then some of the current work that is going on in our lab here at Iowa State. So as we start this webinar, there might be some problems throughout Iowa as well as the rest of the US. As summer hits with eggshell quality in terms of heat stress and needing to change diets or reformulate diets based on price. But the truth is that there's no magic bullet to solve all eggshell issues with just one change in the barn. So what we're going to talk about today are some very simple or key areas that we can assess in order to contribute to improving different areas of eggshell quality. So it's likely that you might have to make adjustments in several areas in order to kind of hit the problems that are all compounding that might all contribute to reduced egg quality. So the first, of course, is know what hen strain you have in your barns, which seems pretty simple, but I think people forget over time if they've either changed strains or they've always been using the same one, that the genetics are also changing over time. The second, of course, is nutrition. We have some key management strategies that also might help improve eggshell quality, and then, of course, the sort of caging system that you are working with. So one of the first things I would like to discuss today is actually some more physiology of how the hen is using calcium from her diet and also her body in order to continue laying eggs over the lay period. So we're going to talk just a little bit about the different types of bone that are in laying hens and how this can contribute to increased or decreased eggshell quality. So the types of bone that are in laying hens are much different than other sorts of production animals. So first off, we have pneumatic bone or hollow bone, as you may have heard. It's actually physically connected to the bird's airway, and it serves as an extra space for air when they're taking in oxygen. And part of this is to lighten their skeleton for flight, and it also provides buoyancy. So if you look at the graph or the figure on the right, what we're looking at are the two different types of bone that are in the bird's body. So pneumatic bone would be anything that is situated around the bird's respiratory tract, and also a section of bone on the bird's head. Now, medullary bone, which is the important bone for calcium turnover during the laying cycle, is a very dynamic source of calcium for eggshell during lay. So it is very important that this bone has good calcium deposits that are put on as the bird is a pullet into the beginning of her lay cycle. So underfeeding calcium, phosphorus, or vitamin D as birds are approaching the time point where they lay their first egg can actually be very detrimental. So you might think, well, I'm saving some money on my diet, but overall what you're doing is setting up a laying hen to not be able to lay to her full potential during peak and post-peak lay. So when we think about how this bone is actually used, it's a very interesting process. So the two graphs are showing you how calcium is moved around in the hen's body based on whether it's coming from diet or her actual bone. So importantly, on the graph on the lower left, what we're looking at is the shell ash, or basically the measure of calcium and phosphorus being put into the shell, and then hours past her ovulation. So you can see that the majority of the eggshell is being put on in this fashion where about 6 to 10 hours after she ovulates, she's already formed the albumin layers as well as the thin shell membrane, and then she starts in her uterus putting on the calcium carbonate shell. So what happens is she goes through a period of faster calcium accretion that sort of reduces and then finally tails off as she gets ready to lay that egg. And most of this eggshell calcium is actually deposited during the night when she's not eating. So this means that we have to be very careful about the types of calcium that are in her diet, as well as making sure that she is maintaining medullary stores of calcium in order to access this when she's not actively consuming feed. So if we look at the overall calcium balance in a 35-week-old hen, we're putting about 4,000 milligrams into her diet. Of course, she's not perfect at utilizing all of the calcium or phosphorus that comes into her GI tract from the diet. So there is some fecal and urinary loss, but medullary bone is contributing about 0.1 grams out of the total amount that is put onto the shell. And that particular calcium source can be accessed when the hen doesn't have any incoming calcium from the diet. So something to remember that I've just mentioned is that the most active period for laying down calcium carbonate to the shell is during that dark photo period when she's not eating. The overall calcium reserve in medullary bone is less than 1 gram, but normally the total amount that's coming from that bone is no more than 0.1 of the total 2 grams that are in the shell. So yes, it's not the majority of the calcium, but it is a very important source because of how the bird is adding calcium to her shell. So something very important that you can start to think about is what is the nutrition from my pullets side of the production before that bird comes into a laying house. So if you're underformulating calcium and phosphorus in pre-lay diets in an attempt to reduce diet costs or reduce the amount of phosphorus that's in the bird's excreta, or if you're not switching to a layer diet once you start to see eggs appearing, you could be underformulating overall calcium for what that bird's needs are in order to build up medullary bone and also her overall skeleton. So very importantly, if you are not heavily involved in how your bird diet is formulated, either with a nutritionist, it's something that is very important for you to learn or spend a little bit more time thinking about. So during the pullet stage, we expect them to be fed a finer calcium carbonate, so less than 2 millimeters when we look at the grind size. So coarser limestone, it can be used in a pre-lay diet up to about 50% of the overall diet. And when we say coarse limestone, we're talking 2 to 4 millimeters overall. The reason that this is beneficial is because it takes longer to break down. So overall, the bird has more time to access and absorb the calcium. So it can hang out in the gizzard once other feed may have already passed through and provide a longer or increased time in order to absorb that particular calcium, where the fine calcium carbonate would be absorbed much quicker. And then once the bird has no feed in her system, it has basically no way to access additional calcium, whereas this coarse limestone provides longer duration during digestion. So we don't typically switch So another thing that can be done in order to improve eggshell quality is that trace minerals can be increased from up to 20 to 25 parts per million copper and 100 to 120 parts per million zinc. So a serious issue that can occur is when marginal vitamin D, calcium, or phosphorus are fed. And this is because the bird can continue to lay, but with an absolute deficiency, usually the bird ceases laying within a week or two of the deficiency that shows up. So it's almost worse to have a marginal deficiency because it's something that the bird can continue to kind of put different resources together until she finally cannot go any further. So marginal calcium in the diet will force reliance on medullary bone and reduce shell quality. So even though only about 0.1 grams on average of that 2 grams of calcium is coming from medullary bone, that's in a situation where the bird's diet is replete in calcium. So if the bird's diet is not, she can pull out more calcium from her medullary bone, and that causes a faster decline in the bird's condition. So with vitamin D deficiencies, typically the issue is it's either underformulated or something has happened to the actual vitamin D where the quality is reduced, such as improper storage or interactions with other parts of the diets. So we end up with a dramatic loss in shell weight. So you can see that these two together both can cause eggshell issues. And part of the issue with a vitamin D deficiency is vitamin D aids the absorption of calcium from the diet. So this is a compound issue when there's a vitamin D deficiency in the diet. So here are some examples of calcium, phosphorus, and vitamin D using phase feeding over the production cycle of a laying hen. So this information is from Highline, and they are a good resource for all sorts of different strains of laying hens. So here what we're looking at are the calcium, the available phosphorus, and vitamin D in terms of the age of the hen. So the important thing to note about phosphorus is that we're not considering total phosphorus. We are considering the available phosphorus that has either been made available through the use of a phytase or inorganic sources that are added to the diet. So in the growing phase of a pullet, the ratio of calcium to phosphorus is about 2 to 1. So 2 parts calcium to 1 part available, not total phosphorus. Vitamin D is usually set at a standard amount, so that's not changing over time. But what you can see is that in this pre-lay diet, what is increasing is calcium has started to double from the grower phase. So you can see how this might be beneficial in order to continue to develop medullary bone and the entire skeleton before the bird is being asked to use calcium towards an egg instead of her skeletal growth. So when you see or start to see eggs appearing in your flock, the first egg to peak, that bird is getting about 4 to 4.2 grams of calcium in her diet per day. So you've seen now we're looking at, instead of expressing as a percent, we're expressing as a total grams that are added to that bird's diet. And the reason that this is important is because the temperature of the barn or her ambient temperature can change the amount of feed that a hen eats. So we need to make sure that we are either diluting out or concentrating the diet so she is getting that 4 grams of calcium a day to maintain optimal production. So as the bird's body is being taxed over time and she's getting past that peak lay period, we're actually increasing calcium because she has been using medullary bone and in some cases has not been able to replace. So the other important part of this figure is that we're looking at the particle size ratios of calcium carbonate over time as we're feeding in these diets. So as I mentioned before, pullets are going to receive very fine calcium carbonate up until that pre-lay diet where we can add about half as that coarse calcium carbonate, and that is because we want that to be released slower. So as that bird ages, we're increasing the coarse percent of calcium carbonate in that total ratio, and this is to help support her lay, especially as she's getting in that post-peak period. So something else, of course, to monitor in your flock is what are the diseases that are present. And of course, some of these diseases are going to depend on the sort of caging that the birds are being kept in, as well as the final market that the birds are going to be, the eggs from the birds are going to be sold to. So we have two very good poultry vets here in Iowa. Sudden flock changes, increases in mortality or condition of the birds, either Dr. Yuko Sato or Dr. Mohammed Al-Ghazar are great resources to call at the ISU Veterinary Diagnostic Lab, and they can easily help you determine what is going on in your flock. But some example diseases that are going to change production of your hens, infectious bursal disease, of course, in pullets causes high mortality, so they don't make it to the layer stage. In fact, some respiratory diseases like infectious bronchitis, they damage the uterus or the eggshell gland. And then, of course, Newcastle can contribute to reduced production. So there are some genetic strains that have been noted to have differences in eggshell quality over time just due to the strain. So it's known that the Highline W36 has some genetic issues with eggshell quality just as birds age. And this wasn't a problem in the past when birds were not molted. So genetically, what these birds are doing is when they hit about 55 to 60 weeks, the birds have reduced shell quality. And it used to be not an issue just because the birds were molted. But now, since we're pushing production out much further than 60 weeks, it's something that needs to be taken into consideration. So the great geneticists, they understand that this is a problem and they're of course working on it, but sometimes a decrease in eggshell quality may have nothing to do with management. It could be the genetic strain of the bird, and then we need to use the diet and management strategies to support that or improve it. So one of the most important things that you can do besides fixing or improving the nutrition in your barn is to just go into the barn. It seems a little silly or trivial, but if you've just recently made an adjustment with your nutritionist and you've changed the ration that the birds are eating, you need to go into the barn, watch their behavior, and also their feed intake. So something to notice, are the birds hot? Are corners, ceilings, or end of buildings created? Are there hot spots there during the middle of the summer? So you might have a perfect conditions at the front or the center at ground level in the barn, but once you start going up catwalks, are there places where the temperature is obviously different? That's something to really note in your barn situations, and change your management and your ventilation so you can try to keep as consistent of a temperature as possible. You have to listen to the birds. Again, it seems pretty trivial, but they're going to tell you what is and is not working. Another thing to monitor is whether or not the bird is consuming the recommended intake for its age, because if it's not receiving adequate calcium or phosphorus. So heat stress is something that we have to combat here in Iowa over the summer. And one thing that a bird might do when it's hot is it might reduce the amount of total feed that's taken in. So if we assume the bird's intake to be 95 or 100 grams, and the bird is suddenly eating maybe 75 or 80, what we need to do is concentrate the diet down to assume an 80-gram intake, and then we can assure that calcium and phosphorus are being met. So that is something to watch. The weather, basically, you can work with your nutritionist and feed mill to determine whether or not this is something that is feasible for you. If you see a couple of weeks of heat stress that might be coming on due to warm temperatures, what you can do is work with your nutritionist to perhaps put together a concentrated diet that might work based on the number of bins you have and what your feed mill is willing to do in terms of working with you and changing regular formulation. So monitor what is left after, if you've ever changed the diet or there's an environmental stress. Is there feed left in the feeders, or are the birds consuming it faster? And then, of course, water intake. Making sure that sodium levels in the water are not terribly high and the birds are continuing to drink water. So if you notice that the weather is going to be hot, other things you can do besides increasing or concentrating the diet are increasing trace minerals. Ensure that you're using the larger particle calcium, at least half of it during peaking, and increase that. So this would be something to talk to your nutritionist about to ensure that this is something that they're typically doing. Okay, so just to summarize what we've talked about in this first section, observe your flock for behavior and uniformity. Make sure you're weighing birds and determining uniformity in different regions of your barn. Make sure that the pullets are coming in with adequate bone, that they've been moved to this pre-lay diet that has increased amounts of calcium and phosphorus. This will pay off in the long run. Monitor nutrition and feed intake, so optimize your diet where deficiencies or marginal deficiencies might exist or as the weather might require. Find the temperature hot zones in your barn, monitor your water intake, and of course, account for the strain and your expected production goals. So these are all things that might be small in their own, but can kind of add up to cause some issues that might reduce eggshell quality. So now that we've sort of talked about some overall changes that can improve your production, I'm going to spend a little bit of time talking about a few different ways that you might be able to either have a value-added egg product or use some common byproducts that are found in Iowa. So the first thing we're going to talk about today is adding omega-3s to your hen diet in order to produce a value-added omega-3 egg. So of course, this is something that you would have to have in contract form if you're expecting to sell these eggs at a additional or premium price. But something interesting that my lab is looking at is, so we have this value-added product for human health, of course, where we've concentrated omega-3s in eggs, but is there also a dual benefit to the bird through increasing the feeding of omega-3s in order to get her to enrich an egg? So there are two different sources that are mainly used in poultry production in order to get added omega-3s or enriched omega-3s in an egg. So the first is fish oil and the second is flaxseed. So flaxseed is a much more common ingredient than fish oil. Part of it is due to cost and also how much can be formulated into the diet before eggs have an off taste. So flaxseed consists of 18% alpha linolenic acid, or ALA, and fish oil contains 18% eicosapentaenoic and 12% docosahexaenoic, or DHA. So all these three fatty acids are all considered omega-3s. However, they have different biological properties. So the flaxseed that we're looking at on the left has predominantly ALA, and ALA is a precursor to the types of fatty acids that are in fish oil, EPA and DHA. So EPA and DHA are ones that you may have heard of as far as supplements of fish oil for human health. These are the ones that have the positive beneficial effects in terms of reducing inflammation, etc. But not to say that the fatty acid on the left is not good, but it doesn't have as many positive beneficial effects because, of course, it's a precursor and it has to be changed from ALA, it has to be saturated, desaturated in order to get EPA and DHA. So there is this consumer demand for omega-3 enriched table eggs. So some of the common fats that are added to poultry diets include flax oil, soy oil, and corn oil. And of course, the omega-3 content of flax is the highest, and it's pretty low in soy and corn oil. So for those of you that like to understand the metabolism of these lipids and how corn oil acts in an animal's body versus these omega-3s, so omega-3 and omega-6 fatty acids both compete for the same rate-limiting enzyme, a delta-6 desaturase. So if you have increased the amount of omega-3s in the diet, that means that at the rate-limiting step, an omega-3 can replace an omega-6. And while this is important because downstream products of omega-6 fatty acids end up having pro-inflammatory properties. So certain inflammatory cytokines, reactive oxygen species, etc., are more likely to be produced from an omega-6 versus an omega-3. So as you can see here, I was saying that the beneficial effects are not necessarily from ALA, but of course the delta-6 desaturase and an elongase has to convert ALA to EPA, which is then converted to DHA. So these are the beneficial fatty acids that give beneficial effects when we feed omega-3s. So we conducted a trial. It's published if you are interested in looking at the full published trial. It's on the lower left of the slide. But what we did is we looked at different forms of flaxseed that can be added to the hen diet in order to increase the overall concentration of omega-3s. So we're looking at total flaxseed oil concentration from two different sources. So MFS is milled flaxseed and FSO is flaxseed oil. So they were balanced based on the oil content. So we added either 1, 2, 3, or 5%. And then on the other axis, what we're looking at is the overall omega-3 fatty acids. So ALA, DHA, and EPA. And what we can see is that compared to milled flaxseed, the flaxseed oil deposited about 2 times as much total omega-3s in the diet. So it seems to be a pretty efficient way of adding diet. And flaxseed oil is not as common of a inclusion for getting increased omega-3s as milled flaxseed is. So part of the reason that milled flaxseed didn't perform exactly as well as flaxseed oil is that milled flaxseed is the entire seed that's been ground, whereas flaxseed oil is just oil that is extracted. And so the overall total amount that we're adding of these products to the diet was based on the oil concentration in each product. So we also looked at the transfer of EPA and DHA to the egg yolk, and we found basically as we increased the oil concentration, there was no change between either product, and this is expected because remember that flaxseed is mainly made of ALA. So in this study, the laying hen study, we found that the flaxseed oil was well tolerated, it's energy dense. The body weight gain was not detrimentally affected by using flaxseed oil, and we doubled ALA deposition as compared to milled flaxseed. So it could be an alternative for ground flaxseed. It may be more cost effective, and there might also be a dual benefit to the laying hen. So after this trial, we took laying hens that had been fed omega-3s and we induced inflammation by giving them an LPS challenge. So that's a part of the bacterial wall that the immune system recognizes as foreign. And what we found was that birds that were fed an omega-3 diet had less inflammation than birds that were not. So that could be an overall benefit to the bird as well as an enriched food product for humans. So of course, feeding omega-3s is something that is based on the market that you're selling your eggs or your meat product to. But I'm going to now talk about distiller's grains, which is a byproduct that can easily be used in both laying hen and broiler diets. And this is a decision that can be made easily by a producer in conjunction with his nutritionist. So overall, the feeding value of distillers grains is in the apparent metabolizable energy, the amino acids, and the phosphorus, or the available phosphorus content. So we're going to talk about how do we determine the feeding value of distillers grains. And the reason we're doing this is because there happens to be a wide variety in the methods of producing distillers grains, as well as how much oil is taken out of these. So when you are asked, well, do you want to add these distillers to your diet? We can offer you some different ways to determine how useful they can be in your particular diet, so you ensure that you're meeting energy requirements and overall amino acids and phosphorus if you happen to substitute something else out to use these. So option one is to use a prediction equation, and this is several different groups have done these, and they've put together a few that could be of use to you. And then option two would be to run a bioassay, which is not feasible for every lot of distillers grain, but we'll describe how these both can be used together to improve the use of distillers in a diet. So distillers have a good fatty acid profile. They contain over half of their fat is linoleic, which is a required fatty acid for the bird. They're compatible with all vegetable diets if that is something that you're required to follow. And they have very bioavailable carotenoids. So basically we can use them as a way to make a darker yolk. So there was a study that was done in 2016, and they found that the carotenoids in distillers corn oil were 93% bioavailable, which is a very good number, compared to a commercial marigold extract. And distillers grains that have been purged of their oil or most of their oil content, they found in a separate study that they contain highly bioavailable xanthophylls to increase skin yellowness. So in a bird that's being sold to a market where yellow skin is valued, this can be an added benefit to adding distillers grains to your diet. And in distillers grains, over half of the total phosphorus is available. So that is something that is exceptional because it can reduce our reliance on inorganic sources of phosphorus as well as the use of phytases. So here what we're looking at are different average NRC values for these different components of corn and distillers grains. So we see that corn itself has 0.28 on average percent total phosphorus, and distillers has 0.71. And we can see here the value of distillers grains is that they contain a significantly higher amount of available phosphorus. So as we go through these different values, one thing to note is that the rule of thumb is that from corn to distillers grains, anything that is in the corn is going to be about 3 times as concentrated in distillers grains. So as we look through the fat values, this holds true, as well as lysine, methionine, and threonine, which are the most limiting amino acids in poultry diets that are based on corn and soybean meal. So they're a nice source of these additional amino acids because they're more concentrated as compared to their parent source, the corn. Now, this also means if we have a year which is heavy in mycotoxin load, that the mycotoxin is also going to be 3 times as concentrated. So it's something just to think about. It's not something to deter you from using distillers grains, but just be cognizant that the ethanol distillation process does not rid the corn of the original mycotoxin content. So when we determine how valuable a feedstuff is for poultry, what we're doing is we're determining the amount of metabolizable energy. So the way that we determine this is we take a feedstuff and we put it in a bomb calorimeter and we literally blow it up. So we get a gross energy value from the amount of heat that is produced when that particular feedstuff is put into the bomb calorimeter. And then we account for energy that is lost via the bird's excreta, which in this case is feces and urine. So the way that we determine this metabolizable energy, first we can determine it by a process where we feed we precision feed an animal an amount of an ingredient after it's been fasted, and then we collect its excreta and we determine how much metabolizable energy is left when it exits the bird. And then another way to do this is we can determine apparent metabolizable energy. So we put the ingredient of choice formulated into a diet at a very high percent, let's say 50 or 60% of the overall diet. So in this case, it's not a fully formulated ration. We're just trying to test this one particular component. And then we can either collect excreta or we can sacrifice the birds and collect an ilio sample. So you'll see on the bottom that these values, either true or apparent, they vary just a little bit. But when we compare conventional to reduced oil distiller's grains, we kind of get an idea of the overall metabolizable energy value for these different methods of measuring how valuable that feedstuff is to the bird. So you can see in this composite apparent metabolizable energy by feeding a high amount in the diet versus total, the values are a bit tighter when we only feed the single ingredient versus feeding slightly less formulated diet that also contains a high amount of that particular feedstuff. So of course we can't run these bioassays all the time because they're expensive, and there are many lots of distillers in the US. So these different groups have created prediction equations that use chemical analysis of the feedstuff in order to predict what the feeding value might be. So an important thing to note is that the first group, they used they based their prediction equations on a range of co-products, of distillers' products. And then the second group predicted the energy based on the oil content. And then the third paper from the same group had predicted in actual values, so actually run through bioassay versus the prediction equation. So the reason to understand how the authors got their different values and based on what their ingredient was is very important. So Rochelle et al. Used distillers grains as well as 2 high protein distillers products. So if we look at their 2 equations that they used in order to predict energy value, the values that you would need to know from your particular product would be ether extract or fat content. You would need to know the ash content, as well as hemicellulose. So that's our first equation. Now equation 2, if you're not able to analyze for hemicellulose, if the lab you send it to or the particular feed mill doesn't have that, we can use NDF, GE, and crude protein. So these, based on what values you have for your distillers grains, can help you analyze what might the value of that lot be. So this group had prediction equations based on a range of fat contents in the distillers grains. So their prediction equations require GE, TDF, crude protein, and starch values. Their fourth equation excludes total dietary fiber, so they're using GE, crude protein, starch, and NDF. So you can see that based on which equation you might use, you've got to have different analytical values. So if we take all of those different methods and we compare them, what we can see is that each equation can change based on the chemical, can change in its accuracy based on the chemical composition of that particular distiller's grains. So as we look through these 15 samples, we can see that there are some over and some underestimates of the actual values from the distiller's grains in terms of apparent metabolizable energy. And some of them kind of were spot on with the average. So it's important to understand, is my product a high protein distillers? Is it reduced oil, etc.? And then you can kind of have some different measurements to get that average. What they also found out was that there, of course, now with value-added products taking oil off of the distillers, overall, distillers can range from about 2.5 to over 14% oil. So that's something to note if you are thinking about using a distiller's product. And also that the crude fat is a poor predictor of the apparent metabolizable energy. So this group found that the most accurate prediction for apparent metabolizable energy was to calculate digestible energy and then use digestible to calculate metabolizable energy. So they found that that was, in their case, the most accurate equation. But we can see that from these different samples, we have an expectation of what the values should be, and then we can see actual happens to be all over the place using crude fat as the predictor. So if you're considering using the co-product distiller's corn oil in your hen diets, it's a pretty nice fat source because it does a few different things. So it happens as compared to refined corn oil, it's almost the same in digestibility. Unless there's a high percent of free fatty acids in the diet, then the digestibility is significantly reduced. But it's pretty spot on as far as the apparent metabolizable energy. When we look at the amino acid content of these distillers grains, here we're looking at values that are published in the NRC from 1994 and then 2 different suppliers. And we can see that some of these values kind of vary, and then other ones have a larger range of possibilities. And part of this is due to the quality of the corn going in, and then some of the different process variations. So it seems on average they're about 27.5% crude protein. These particular products before corn oil, or the distiller's corn oil was heavily extracted, ranged around 9% fat. Lysine was between 65 and 70%. Methionine was pretty close, 84 to 86, and then threonine was pretty standard for all of them. So one thing to note is that even though there are different or higher levels of these amino acids in the distiller's product, we want to make sure that these are still digestible to the laying hen. So what we're looking at here are, this was a study that was done in 30-week-old laying hens. We've got 5 different distiller's products and then 3 different corn products. And overall, it looks like between lysine, which is in the light blue, and then methionine, which is in the royal purple, and then threonine, which is in gray. Overall, the digestibility is slightly reduced based on these distillers grains as compared to corn. But overall, remember that there are more of these amino acids in distillers versus corn when we consider the same amount. So there may be some slightly reduced digestibility, but there are more total amino acids in distillers versus corn. So for those of you who might be interested in using distillers in broiler production, what we're looking at here are the digestibility of the same distillers grains in a hen versus a broiler for 5 different distillers products. And basically, the trend that we see is that the broiler is typically able to extract more energy out of distillers grains than laying hens are. And I mean, that is totally 100% understandable because the job of the broiler is to grow, so their digestive tracts have a bit more efficient system than the laying hen. But when we look at some of the most limiting amino acids in the diets, in some cases, the laying hens are just as good at digesting those limiting amino acids, and in some cases, they're reduced. So not to say that overall the broiler is better, but in most cases, the broiler is as good or better at digesting the overall amino acids versus a laying hen. So I'm going to show you some data of different percentages of distillers grains being added to laying hen diets. So here we've got either 0 or 15% distillers grains being added to a laying hen diet. And there was one point in production where the distillers grains seemed to slightly reduce the hen day egg production, but overall they were comparable. And there's a case on the lower graph that also might show some time points that are significantly reduced, but overall it seems comparable. So what you as a producer have to do is decide, is the diet versus trade-off of potentially reduced production at different time points worth the reduction in diet cost. But of course, this was just one study, and we don't know much about the overall quality of the distillers grains and how the diets were formulated, et cetera, from just this graph. But formulating up to 15% is not going to tank your egg production. An interesting effect of feeding distillers grains is that when we feed either commercial or low density, so oil extracted, for example, we get an increase or improvement in the orangeness or darker yellowness of the egg yolk. So that's something to think about. That's a good side effect of feeding distillers grains. So if we increase the percent of distillers grains in the hen diet, we linearly increase egg color. So for those of you who are familiar with this Roche color fan, we are increasing numerically the yellowness to the orange scale of that egg. We also improve nitrogen and phosphorus retention, which is a good benefit because we end up losing less in the animal's manure. So that is always something that's great to improve the digestion of those nutrients, so we have less runoff and also less other inputs needed in the diet to meet those requirements. So does adding distillers grains change the egg quality? So what we're looking at here are weeks of age of the laying hen and percent of distillers grain added to her diet. So what these researchers did was they added 0, 17, 35, or up to 50, which is very high in terms of whether or not we would probably not be adding that to practical diets. Excuse me, but basically, what we see is that the shell breaking strength can be improved if we added 50% distillers grain to the laying hen diet, likely due to some of that nitrogen and phosphorus retention. But what we also see is that adding distillers to the hen's diet is not reducing egg strength, so that's great. So if we look at the internal egg quality components, so the color, the Howe unit, egg composition, etc., we see that as we increase the percentage of distillers grain in the diet, we again see this nice trend of getting darker or more orange. And we also see that there is, in most cases, an improvement in overall storage time or protein quality of that egg. And we don't significantly change the egg weight components, which is great. The one thing we do change is we improve the shell over that 50% distillers grains, but again, that's not likely a practical formulation. So some of the things that we're currently doing work on here at my lab at Iowa State. So we work with both laying hens and broilers, and one of the things we're doing is trying to use components that from different feedstuffs that could be concentrated to provide beneficial anti-inflammatory effects to poultry. So right now we're investigating some different bioactive components in alfalfa. We have some very cool projects going on that look at the welfare and behavior of broilers, so improving leg lamenesses, adding to and improving guidelines for auditing that producers can do on their own, so they can do self-audits, etc., so they're compliant with the different requirements for the companies that they grow for. Another great thing that we do is we try to understand the immunological effects of feed additives. So we like to put science or understanding behind how some of these different feed additives work. And one way that we do that is we look at the changes in immune cell function based on housing, age of the bird, strain of the bird, etc. And then we have a very cool collaborative project going on with the USDA regarding how the hen's bacteria colonizes eggshells and how that changes the microbiome and the immune system development in the chick that hatches out from that particular shell. So a very cool part of the work we do here at ISU is we teach undergrads about the poultry industry, so they have a great appreciation of more animals that happen to be popular here in Iowa, as well as potential jobs in the future. So one of the things that we do in my lab is we compare the immune response between broilers and laying hens. And of course, broilers have a short production time, whereas layers, we can kind of see a memory response because their lifespan is significantly longer. So one of the things that we do is we isolate immune cells from the blood of these birds, and we see how they're changing over time based on either a housing condition or a feed additive that we have given the birds. And the reason that this is important is that the immune cells, based on what fuel they're using, have different tendencies either to be inflammatory or not inflammatory. So one of the things that we can measure is how much oxygen are the cells using in a baseline way, and then how is the pH of the media changing based on the pathways that they're choosing. So when an animal is in, or the cells are in a more inflammatory state, they tend to go towards anaerobic metabolism or glycolytic metabolism, which is less efficient. And then when they're kind of in a resting or more quiescent state, they're using mitochondrial respiration. So they have the option of using glucose or carbohydrate, amino acids, or lipids. And one of the very cool things that we found was that the ability or the choice to use mitochondrial respiration is different between broilers and laying hens. So that is something that just on a baseline way that we are continuing to explore, whether it's an age-related, a housing conditions-related, sickness or disease, etc. And the reason that this is important is because then we can develop precision nutrition based on the type of support that a particular immune response might need. So that's the downstream goal of our lab, is understanding how we can change a bird's diet to best support a challenge that it is particularly undergoing at a time. So here our outcomes are we're training both graduate and undergrad students to be the next great minds. Someone's got to replace us. We're trying to provide precision nutrition based on an immune response and a challenge. We're trying to determine the effect of new housing systems on how the immune cells decide to use different sorts of fuels. We're trying to enhance the immune response that based on dietary components. And then understand how these feed additives affect immune response and overall performance of the bird. And then, of course, we have many industry partners that we respond to industry needs for research, and we are very excited and happy to do so. So thank you guys for spending an hour with me talking about poultry. I hope it was beneficial, and of course, if you have questions, my email is at the beginning, and I would be happy to answer them as best that I can.
Well, thank you so much, Dr. Bobeck, for joining us. And we appreciate you taking the time out for the rebroadcast. If you do have any questions, please direct them to the doctor, and she'll be able to answer them. And we appreciate you taking time out of your day and listening to this broadcast. Thank you so much.